Well, thank you very much. So I'm going to change focus from where we were yesterday and really begin to go back to the basic biology of metronomic therapy and then application to the clinic and what I think are still some of the major drawbacks in the way we approach these therapies. I don't have any stocks or employment with companies, but some of the things I'm going to talk about I do have preclinical trial agreements with. So what I'm going to do is really start this process of beginning to go back to the basic principles of understanding the preclinical data on which metronomic or anti-angiogenic chemotherapy were based, and then talk about it in the context of a couple of actual trials, both completed and those that are ongoing, and then just finish off with what I think are some of the problems that will make the interpretation of these data difficult, and something that I think as a community we're going to have to come together to figure out. Otherwise, we're just going to keep doing these phase 2 trials over and over again without really developing a definitive answer. So I think it's not, I think in a group like this, we're relatively comfortable with the concepts of angiogenesis, in this case a tumor where you can see the clear neovascularization. So I don't think that is so much the question. I do think that we haven't quite agreed on exactly what we mean by metronomic therapy, and I suspect that there is still a lot of heterogeneity within the room. So what I decided to do was go back to, this is a slide that actually Bob had showed yesterday from Judah Folkman and Tim Browder's original work, in which they began developing the definitions, again based on this concept of metronomic, or what they referred to as anti-angiogenic therapy, was therapy that targeted the neovascular compartment. And it really is epitomized by these images. This is the corneal eye assay, where you take that little white dot and basically you implant it into the cornea of a mouse or a rabbit, and then you start to see this dramatic induction of neovascularization, and the height, the circumference, and the density of those vessels is a kind of determinant of the potency of the molecule you use, VEGF, basic FGF, IL-8, et cetera, et cetera. And then on the right-hand side, you can see an animal that has been treated with the standard dose of cyclophosphamide, the kind of human equivalent of 1 gram per meter squared, and you can see that there's significant inhibition in the ability of these cytokines to induce neovascularization. These animals don't have any tumor on them. This is just a pure measure of angiogenesis and the ability of traditional chemotherapy to inhibit neovascularization. And it was out of this principle that the first concept of chemotherapy can be anti-angiogenic really developed. And then you kind of saw this also briefly this morning in the context of the way the experiments were initially done. So on the most left-hand side, control animals, basically if untreated, the tumors grow and kill the animals. If by contrast you do what we've been doing in the oncology clinic for the last 60 years, so this is where the animals were getting cyclophosphamide at the MTD dose of the human equivalent of 1 gram per meter squared, 3 days in a row. It requires a 21-day break period to recover from the toxicity, and you can see after 12 doses, basically all of the tumors have escaped, become resistant, and the mouse dies. And to some extent, this is mostly what happens in human beings. Chemotherapy can slow the tumor down a little bit, but for the most part, they end up escaping and dying. What Tim did was he spent a lot of time developing different methods of giving the drug to see if he could optimize the way in which you weren't attacking the tumor cells, but you were presumably attacking the dividing endothelial cells. And he developed this procedure in which if you gave the same dose of cyclophosphamide every 6 days, it was better than 5 and better than 7. Every 6 days, you had this optimal effect where now only giving 7 doses on an every 6-day schedule worked much, much better than giving 12 doses on the standard way that we applied in the clinic. And this again became this concept of anti-angiogenesis or metronomic therapy, because as you can see, the drugs are given on a regular metronomic-like approach. The other thing he did to prove that this was actually in this case endothelial or microenvironment, is he then took tumors that were super resistant to cyclophosphamide so that cyclophosphamide couldn't kill them, and he basically repeated the experiment and was again able to show the same thing, that you only see tumor shrinkage not by killing tumor cells but by killing the endothelium. And again, it was out of this concept that we really got comfortable with the idea that chemotherapy, when applied in a lower but more regular dosing, could be anti-angiogenic. And again, this was published in 2000, and then immediately afterwards was the information that you've already heard from Bob Kerbel and his group, in which they combined it with a VEGF inhibitor and got actually very similar kind of approaches. Well, as is often true, actually starting going back to 1960 and going forward, the literature is absolutely rife with articles in which patients that had relapsed after standard chemotherapy and were being sent home to die were being given things like oral etoposide. In fact, I was giving oral etoposide from the day I started as a clinician in the oncology clinic with no understanding about why it worked. This is a paper from 1996, David Ashley, in which he took 7 kids with relapsed medulloblastoma, all of whom had received high-dose intravenous etoposide or VP16. Tumors had become resistant, and for reasons that were never exactly clear, other than we just all did it, we would then send those kids home on oral etoposide, the same drug they'd already proven resistant to, and let them die. And what they had identified in this particular paper is that 6 out of the 7 kids had a dramatic response to the oral etoposide given daily at 50 milligrams per meter squared. And you can kind of see that here compared to what is on your left-hand side. Lots of areas of tumors. The numbers don't show up particularly well, but if you look, you can see them. And on the right-hand side, you can now see that most of the tumors actually disappeared. Now, eventually, all of the tumors come back, and all of the children did end up dying. But it was this dramatic response that raised the question of mechanism. And obviously, with the work that Tim and Judah were doing, we began to think that maybe we had an understanding of the mechanism of why these responses were occurring. Now, Tim had not done any of the work on etoposide. All of his work had been on initially cyclophosphamide, then adriamycin, and a number of other drugs. So in my lab, in conjunction with Deepak Panigrahi, whose work we talked about yesterday, we performed what are the kind of typical anti-angiogenic assays. For example, the endothelial proliferation assay, in which if you use increasing doses of oral etoposide up to 1 micromolar, you can inhibit the proliferation of endothelial cells. This doesn't actually kill any of the endothelial cells. You need about 10 times a higher dose to actually start killing cells. Similar for the tube formation assay and again the corneal eye assay. This is kind of proof that at the kinds of doses that you get in human beings with oral dosing of etoposide, you can actually get inhibition of endothelial cell function. Remember, none of these assays have anything to do with tumors. This is just the inhibition of angiogenesis. Obviously, you can do the experiments in a variety of tumor models. These are just a couple of examples: Lewis lung carcinoma, fibrosarcoma, glioblastoma, where again, in daily oral gavage of these animals at either half the dose or full metronomic dosing of what is still low dose etoposide, you can again get good tumor response, even in tumors that have been made resistant to high-dose intravenous VP16. And again, this has been published, so I won't go through it in more detail. Now, at the same time, Judah's lab had become interested in other molecules that might also have the ability to inhibit endothelial cell function. And so one of the postdocs in the lab, Rob D'Amato, started a project where he started to scour the internet to begin to look for drugs that had caused certain developmental defects in children. And one of the ones he discovered was this condition called dysmelia in children, in pregnant women that had received thalidomide, in which the arms, in particular, of the developing babies basically grow right out of the shoulders. There's no actual forearm that's made. And it turned out the reason that you don't make a forearm in the context of thalidomide during day 35 and 50 of development is because the vascular bud that needs to develop to create your arm never forms because it's inhibited by thalidomide. What he also discovered is that thalidomide is a potent downregulator of both basic FGF and VEGF, which back in 1994 we assumed to be the major angiogenic cytokines that basically regulated all angiogenesis, obviously something we now know not to be correct. At the same time, there are also multiple groups that have been working on the role of Celebrex, particularly in colon cancer, but the recognition of COX-2 activity in endothelial cells. You need COX-2 in order for endothelial cells to divide, so it would make a good compound in which to inhibit endothelial cell proliferation. And obviously, as we looked at those 4 oral drugs, we put together a clinical trial. This was for children in terminal care, again asking the questions, could you modulate some of their disease status using these 4 oral compounds? It's oral so that they can go home, they don't have to come to the clinic. We understood the mechanism. It's actually you're on 3 drugs at any time. You're always on thalidomide, you're always on Celebrex, but you alternate the VP16 and the cyclophosphamide because we know they can induce their own auto-degradation, and we didn't want to allow the tumors to do that. So you get 21 days of one drug, and then you immediately switch over. There are no breaks between them. And the idea was, our hope was that under those circumstances, we would be able to get some of that anti-endothelial activity. So in this pilot trial, we basically just took 20 kids with relapsed incurable disease, virtually all of whom had already had the MTD doses of both cyclophosphamide and etoposide, and we basically started them on this home regimen. And this experiment, between the time that it was proposed, that we got FDA approval with the IND, actually accrued and treated the patients, took 5 years. So for those of you in the lab that complain about how slow your mouse experiments are, for those in the clinic will understand how much more difficult it is to kind of do these experiments in a reasonable fashion. Just very briefly, the results. So 8 of the patients just had their tumors just continued to grow. It's as if they were getting nothing. In fact, some of the patients came off the study within 2 days of starting because of rapid tumor growth. Whether any metronomic therapy would work within 2 days, we could certainly debate. Interestingly, there were 4 patients who actually had some slowing of their tumor growth. They basically remained stable for 3 to 6 months before the tumor finally took off, and whether that's of clinical meaningful is obviously, again, something we can debate. But what was particularly interesting is that 8 patients actually had complete arrest of their tumor and or shrinkage, and 10 of the 20 patients had actually ended up being on this metronomic therapy longer than they had on the prior therapy that got them into this trial. And in fact, it was so well tolerated that of the 8 patients that actually remained for the full therapy, 7 of them refused to discontinue therapy once the trial was over. And for those of you that have ever treated anyone in the clinic with ICE chemotherapy, when was the last time an ICE chemotherapy patient asked you to have more after the trial is done? It's trouble just getting them to complete the therapy as it is. So we certainly seem to be meeting the criteria of tolerability. We were obviously interested in whether this would affect growth, and it turns out we had 3 young children between the age, basically under the age of 3 on the trial, where growth would still be rapid and therefore easily measured. This is an example of a child that had been on the regimen for almost 3 years, and you can see that the growth parameters stayed unchanged. We had 1 female that was menstruating at the time of entry to the trial that became amenorrheic. That's a well-known toxicity of thalidomide, and so it wasn't a surprise. So this is the Kaplan-Meier curve, again, made up of the rapid progressive patients, that intermediate group, and then those patients that ended up having prolonged stable disease and or response. And the question is obviously, how does one interpret that in the context of 20 random patients, all different diseases, all different prior therapies? Obviously, you can't make much of the conclusion other than it was well tolerated, and you probably need to think about it a little more. And this has been published, so I won't go into any more detail about it. The one interesting thing is we had done a wide biomarker screen as well, and we did identify 1 biomarker. Now, obviously, since we were looking at everything just randomly, we could have found something, but at least it was an exploratory discovery association that the patients that had elevated thrombospondin at the time of entry into the trial were the patients that were most likely to be the ones to respond to the therapy. We actually have tried these therapies and multiple others in a variety of adult tumors, so we tried to look at it in different circumstances. Actually didn't see a lot of activity, and we'll talk about that in the adult trial momentarily. So obviously, while that 5-year trial was going on, we were still in the laboratory asking some important questions about could we identify other drugs on which we could identify some of the same circumstances. And as I had mentioned briefly yesterday, we had been particularly interested in PPR gamma. The PPR gamma pathway is known to be particularly important in endothelial cells. They upregulate the pathway just before they go through cell division as a way of providing the environment on which endothelial cells can appropriately divide. And obviously, because of type 2 diabetes, there are a ton of PPR gamma agonists that actually shut down the pathway. And we therefore actually started some early clinical work with those, discovered that everybody was becoming hypoglycemic and starting to faint, at which point we realized we were never going to be able to do that trial. Fortunately, we had been doing some controls with PPR alpha, which happens to be the regulator of cholesterol, and it turns out that in order for endothelial cells to divide, one of the things they have to do is incorporate new cholesterol into their membrane so that when the membrane divides out, that they can spread it out accordingly. And so we asked the question, would that be an appropriate target? Because again, there was enormous experience with anti-cholesterol drugs, and you've already heard a little bit of that earlier today by some of the other speakers. And the question is, would that be an appropriate target? Well, here are some of the same anti-angiogenic assays, the aortic ring assay, again, dose-dependent inhibition of vascular sprouting, the tube formation inhibition assay, the corneal eye assay, et cetera, in which, again, we were able to show that these PPR alpha agonists were able to inhibit angiogenesis in those models. And again, if you do the kind of standard in vivo assays, again, treating mice, in this case with glioblastoma, with the statins, these PPR alpha agonists, you can again significantly inhibit tumor progression, suggesting that they would have some value. Obviously, without going into the details, we spent a lot of time in the laboratory making sure that we understood the mechanism, and one of the key experiments was we basically made mice that had knockout of PPR alpha-dependent signaling, and then we developed both tumors in those backgrounds as well as in wild type. And you can see in the different constellations here, when the tumor and the mouse are wild type, the tumor grows wildly. If you now treat with a fenofibrate, a PPR alpha agonist, you can dramatically suppress tumor growth. If the tumor doesn't express PPR alpha anymore, and you treat with fenofibrate, you can continue to treat it, presumably because the target is not in the tumor. By contrast, if the microenvironment is negative, even if the tumor is positive, now suddenly fenofibrate doesn't work. And again, that's exactly what you would predict for a drug that is not anti-tumor, it is anti-microenvironment, i.e. targeting the endothelial cells. And it was based on these results that we were convinced that that was the mechanism of action. And again, these papers have been published, so I won't go into that in more detail. We then started to take all of those combinations in a variety of different patterns and ask if you used each of the doses in mice at subtherapeutic levels, could you synergize them to actually begin to show some activity in these multiple combination drugs, on the recognition that we weren't really going to be going forward with single agent compounds. You'll notice the one drug missing here is thalidomide. Remember that the problem with thalidomide, it's not metabolized in mice the same way it is in humans, so mice don't get the dysmelia that humans do. In fact, because mice don't get it is why it was never picked up until human beings started to get it. And it was based on that the FDA now mandates that if you're going to do any human testing, you have to have tested in 2 completely different types of pre-animal models to move a drug forward. So we can't test thalidomide, but in fact the human experience had already done that for us, and it was based on all of this data that we now put together the 5-drug regimen, which is obviously the 4 prior drugs with the addition of fenofibrate. There have been about 100 million prescriptions for fenofibrate written in the United States for patients with hypercholesterolemia, so it's one thing we know a lot about. The only major toxicity is in patients with high cholesterol level. You sometimes get cholesterol gallstones when you start to decrease those levels, something we weren't worried about in a terminal cancer population. So this was a prospective trial of 101 consecutive patients with incurable disease under the age of 21. The majority had already received the maximally tolerated dose of both etoposide and or cyclophosphamide, and that they had to have evaluable disease. So they were started on the therapy, and again, this clinical trial took us another 5+ years to complete. So this was an 11-institution trial within the United States. And what we had done was we were beginning to recognize that the same anti-angiogenic therapy was not going to work in all circumstances. It looked like each set of tumors had a different way of inducing the microenvironment that might make it more or less susceptible. If, for example, your tumor is upregulating a lot of IL-8 to induce angiogenesis, then a BRAF or VEGF inhibitor may not be the exact approach. And we began to recognize that maybe that would be dependent on tumor type, and that's why we split it out in these. And it was a modified Simon two-stage design. We would accrue 10 patients. If there was evidence of activity, we'd keep accruing. If there wasn't evidence of activity in any cohort, we would stop. These are just the doses of the way the therapy was provided. The goal was to get 6 months of therapy and then basically assess to see how you were doing, given that, again, this was a palliative terminal population. So toxicities were relatively as expected for the drugs. Remember that because most of these are terminal patients, many of them have had craniospinal irradiation, bone marrow transplant with high-dose chemotherapy, so they're already somewhat sensitive, but we got basically all of the side effects that one would expect, but not really anything major. Out of the 101 patients, only 3 patients decided to discontinue therapy due to toxicity, and there were no treatment-related deaths. So this is kind of the outcome, and obviously, given some of the conversation we've already had in this conference, the very idea that you can lump all tumors together as one is probably a gross injustice, but I did it anyways because obviously this is what we were thinking a decade ago. So for example, when you looked at the malignant gliomas, and remember we had by this point also run now 2 trials in adult malignant gliomas, we basically had 1 PR that was transient, that didn't last very long. So it didn't appear that there was a lot of activity in that population. For example, not a single one of the bone tumors responded. Why that is for osteo and Ewing's, given that they should be just as angiogenic as any other tumor, raises a number of questions, and again suggests that it's the microenvironment is much more complicated than maybe we've given credit for. We saw a lot of activity in ependymomas, similar to what we had seen in the 4 drug. Similarly for the low grades, we also saw it in a variety of miscellaneous tumors that at least suggested that there were in some places we would see activity. You'll notice there are not a lot of leukemia patients because the point is they relapse so quickly they never even get onto the trials. We didn't have a lot of neuroblastoma patients because there was competing trials that included a metronomic component to them that overlapped this protocol, so they weren't eligible. And we had some problems with the medulloblastoma patients because, as you can see, we had 1 CR and 1 PR with dramatic shrinkage of the primary tumor followed by rapid development of leptomeningeal disease and dissemination through the spinal cord and progression. And obviously, since cells growing in the spinal fluid are probably not angiogenic dependent, it looked like we were hitting one compartment and completely missing another. And that will become relevant in a moment. So out of the 101 patients, 1 CR, 16% had PRs, and another 38% had prolonged stable disease for at least the 6+ months of the therapy. And 2 patients ended up not being evaluable for analysis. Reasons for stopping: eventually 61% of the patients ended up eventually developing progressive disease. There were 3% with toxicity and 30% that completed the therapy and went on forward. So these are the survival curves for this patient population. Again, the low-grade glioma group does well, but of course they tend to do well in terms of survival. Also, some activity in the miscellaneous group, the red line, and in the ependymoma group. You'll see again that, for example, the bone tumors, high-grade gliomas, we didn't really see much activity. So these are just separation of some of the curves, again, for the low-grade gliomas, where, again, we were seeing activity that was basically equivalent to what you get with the different therapies that have been used, including vincristine carbo, vinblastine, etc. And then similarly for the ependymomas, again, a relatively good 2-year PFS and OS for this population. And again, some of these patients are still long-term survivors, having never progressed again, having completed this therapy. So we had done, had as a specific aim now the assessment, the prospective assessment of the thrombospondin level, and it was interesting that again, it turned out that the only predictive marker of who was going to respond to this were the patients that started with the highest thrombospondin appeared to be the ones that were most likely to respond from the therapy, but it wasn't 100%. There were a couple of patients with high thrombospondin that didn't respond, a couple with low that did, but the vast majority that responded had high levels, suggesting that if your tumor's already got a somewhat anti-angiogenic environment, it may be that anti-angiogenic therapy pushes you over the edge, and that's why you respond, although obviously there's a lot still to be learned there. In literally the hundreds of other secondary markers that we looked at, again, we didn't find any association, VEGF, basic, FGF, et cetera, et cetera. So in the trial summary, again, it was certainly well tolerated. Risk of second malignancy is always a concern. There's one patient that developed secondary leukemia. That patient had also received intravenous high-dose VP16, so it's hard to know whether it came from the metronomic therapy or their upfront therapy. And we've had another patient just recently develop what looks like is going to be MDS, so it's now probably going to be 2 out of 101 patients over a 10-year period that have developed a MDS-like picture. Both of those patients, as I said, had also received high-dose intravenous etoposide. Obviously, not a good strategy for things like high-grade gliomas or bone tumors for reasons we don't understand. And I think one of the biggest mistakes we made in this protocol was not trying to figure out why when someone didn't respond, what was uniquely different other than the biomarker analysis, which turned out not to be as helpful as we had hoped. Again, we saw the same kind of positive responses with ependymomas and low grades, and then in a number of other tumors, the majority of which were actually in the brain, which raises interesting questions about why most of the responding tumors here are central nervous system, and whether the constellation of drugs here just happened to fit more the milieu of the central nervous system as the reason for response. And then just to finish off, one of the things we've been able to do is, as I said, well, we were running this large 8-stratum multi-institutional trial with the recognition that in the medulloblastoma patients that were responding, we were seeing the response in the primary tumor and then development of progression of disease along the leptomeninges. Irene Slavik and the group in Austria said, why don't we add in some Avastin, something that didn't exist at the time we originally proposed the trial, to try and get better VEGF inhibition, and why don't we add intrathecal chemotherapy to try and prevent the explosion of the cells within the leptomeninges. And so she actually did that not on protocol, but she treated 9 children with relapsed medulloblastoma. And for those of you that treat relapsed medulloblastoma after radiation and chemotherapy, the average salvage rate for this disease is somewhere around 5% with things like high-dose chemotherapy and stem cell rescue. And basically after what had been between 3 and 4 years, we were now seeing survival rates of about 50%. And so based on this pilot data, obviously, since these are selected patients, one can't be sure. So we decided to do a phase 2 therapy in which we would test this prospectively. Obviously, we were interested in doing a randomized trial, but since there is no accepted salvage therapy for medullo, the question is, what do you randomize them to? Because you can't randomize a child with a terminal disease to nothing. And so again, we're in the quagmire where we've now got a single-arm phase 2, and the question is, how are we going to interpret those results? So it's not going to stop us from trying, but again, I think we're going to come back to the same issues that we've dealt with in this meeting over and over again, which is after it's done, we're still not going to be sure whether we're on the right track. Again, the trial is based on an objective response rate and then secondary objectives of overall and progression-free survival. It's again based on a modified Simon 3-stage design. So the first 16 patients, if we have at least 2 responses, we would keep going. If by 27 patients we have at least 5 responses, we would keep going up to 38 patients with the listed type 1 and type 2 error. So far we've got 8 patients. Actually, these slides are a little bit out of date, but at the time of the cutoff, we had 8 patients, 6 in first relapse, 2 in third relapse, 7 of whom had metastatic disease at the time of relapse. So this is an example of one of the kids that started the therapy on your left-hand side with the multiple nodules disseminated throughout the central nervous system that you can see in the first 6 months has now had a complete response. In fact, if you look at all of the patients out of the first 8, 3 of them had a CR. 1 of them had a PR. 1 was too early. Looks like it's going to end up being a PR. We had 1 stable disease and 2 progressive disease. So we actually already have 5 responses in the first 8 patients. Remember that in the stage 3 design, we needed 5 patients in the first 20 odd in order to go to the next level. So we already know that we will now go to the full 38, and that's the trial that's accruing. And I think actually, for example, the Marseille site's about to open this week. These were the planned starting doses based on what we had done, but again, because so many of the patients were heavily pretreated with prior radiation and stem cell rescue with transplant, these are the actual starting doses because we tried to build that in to not make it overly toxic. Basically, the toxicity profile is what would be expected for these drugs, so there's been nothing unusual. Not all of these are actually related to the treatment. Some of them are related to the disease or to, for example, their central venous access, but at least it fits well within the context of what we had seen in the prior trials and what would be expected for the drugs. So in summary, the issue here is that we've now got this heterogeneous group of tumors that I showed you in the 5 drugs, some of which responded well and some of which didn't. We've now got this targeted therapy for medulloblastoma, but for those of you that follow medulloblastoma is now considered at least 4 different subtypes of the disease. So how are we going to relate that into the trial that we're doing? It may be metronomic works better for some subtypes than others, and that wasn't controlled because we didn't even know that at the time. There's no control arm, and the question is, how do we in pediatrics develop a control arm when pediatric patients can't really be eligible for control kind of placebo arms? How do we separate out the individual components? The problem is now we have 8 drugs in this combination. How does one really figure out how important each one of those drugs is as we continue to add on? So we've complained a lot about single agent therapy and all of the problems with expecting single agents to do very much. Now that we're getting so many drugs, it's going to be hard to figure out which ones to keep in, which ones not, as we move forward. So just to recognize the preclinical work that I showed you was mostly done by a student, Emily Green, and again, Deepak Panigrahi, when he was a student and then a postdoc in my lab. These are the people as part of the clinical trial and the preclinical development, and then this was the clinical trial that I presented and the funding sources, and I'll stop there.
Thank you very much, Dr.